There's a puppet on Mars right now. "240 million miles away, sitting in a robotic arm. No one put it there for science. No one put it there for discovery. They put it there for money." This is the untold story of how a bet on Mars became the greatest heist in human history. And why that little puppet knows more secrets than NASA ever wanted you to find out. Inside that puppet might be the answer to whether we've ever been alone in the universe. Most puppets on Mars tell us about volcanoes, impacts, ancient floods, the usual geological story. But this one has patterns scientists can't explain with normal chemistry. Rings of minerals that, on Earth, only form one way: through life. Today, we're going to examine what Perseverance actually discovered in July 2024, and why geologists are simultaneously excited and terrified of being wrong. Now, I've spent the last week reading every paper published on this sample, and I've got to say, the data is fascinating and frustrating in equal measure. If you appreciate this kind of research-heavy content, subscribe. It really helps the channel. Alright, here's what happened on Mars. Let's set the scene properly. Jezero crater wasn't chosen randomly. 3.5 billion years ago, this wasn't a dusty impact basin. It was a lake. Rivers flowed in from the northwest, carrying sediment, depositing layers of clay and silt. Clay is critical here. On Earth, clay minerals trap and preserve organic molecules for billions of years. They're nature's filing cabinet for ancient biochemistry. When Perseverance landed in February 2021, the mission brief was clear. Find sedimentary rocks from the lake era. Drill samples. Store them for eventual return to Earth. For 3 years, Percy explored methodically, analyzed dozens of rocks. Most were volcanic basalt, geologically interesting, but biologically dead. Then in July 2024, while examining an ancient riverbed called Neretva Vallis, the rover's cameras locked onto something unusual, a chunk of mudstone the team nicknamed Cheyava Falls. And when the instrument started scanning, the countdown began. 2 systems did the heavy lifting here: PIXL and SHERLOC. PIXL, planetary instrument for X-ray lithochemistry, fires X-rays at rock and analyzes what bounces back. It maps chemical composition at microscopic scale. SHERLOC, scanning habitable environments with Raman and luminescence for organics and chemicals, uses lasers to identify organic molecules and minerals. The data came back bizarre. First, the rock was definitely sedimentary, fine-grained, formed underwater. Good start. Second, embedded throughout the surface were circular features. Tiny, 200 micrometers to maybe a millimeter across. Each one had a bright center and a dark ring around the outside. The science team started calling them leopard spots. Third, and this is where it gets weird, those dark rings were chemically different from the surrounding rock. They were enriched in iron and phosphorus. The specific arrangement suggested a localized chemical reaction had occurred. Something had reduced oxidized iron in that exact spot. Fourth, the rock contained organic compounds, carbon-based molecules. And fifth, its texture screamed, "I spent time underwater." Now, here's why that combination matters. On Earth, when you see organics plus water plus localized iron reduction, there's usually one explanation: bacteria. Specifically, bacteria that don't breathe oxygen. They metabolize iron instead. Let me explain how that works. Certain microbes, Geobacter is the famous example, live in environments with no oxygen. Riverbeds, deep mud, anywhere oxygen can't reach. They've evolved to strip electrons from oxidized iron, using that energy transfer to power their cells. It's called chemosynthesis. The process leaves waste. Reduced iron gets excreted. When that reduced iron meets phosphate in the surrounding water, it crystallizes into a mineral called vivianite. Vivianite forms rings around the bacterial colony, dark halos of iron phosphate marking where the microbes lived. There's another mineral in play here, greigite. It forms when sulfate-reducing bacteria break down sulfates, releasing sulfide that reacts with iron. Both vivianite and greigite are metabolic byproducts. Life's leftovers. And that's exactly the chemistry pattern Perseverance detected in Cheyava Falls. But scientists don't jump to conclusions, so they tried to find non-biological explanations. Science works by falsification. You don't prove your hypothesis correct. You try to prove it wrong. If it survives every attack, then maybe it's true. So the Mars team asked, could these minerals form without life? Hypothesis 1. Volcanic activity. Maybe hot gases from underground volcanic systems leaked into groundwater, delivering sulfides that reacted with iron. No biology needed. Problem. Volcanic systems leave evidence. Altered minerals from heat, hydrothermal veins, temperature signatures. Perseverance scanned the surrounding area extensively. No signs of volcanic influence anywhere nearby. Hypothesis 2. Extreme heat. At temperatures above 150 to 200, organic matter can chemically reduce sulfates without any biological help. Problem. The rocks show no signs of ever experiencing those temperatures. The mineral assemblages are all low temperature. This area was never that hot. Hypothesis 3. Acidic water. In highly acidic conditions, iron and sulfate dissolve easily. Chemical reduction becomes much more likely without biology. This one seemed plausible. Mars might have had acidic lakes. Maybe that's all we're seeing. Chemistry, not life. But then Perseverance found something that killed that theory completely. Sitting near the sample site was a small green mineral. Olivine. If you know geology, you know olivine is the worst mineral for surviving acid. It's incredibly reactive. In acidic water, it disintegrates almost immediately. Olivine's structure is weak. It's held together by ionic bonds between magnesium, iron, and silicate. Hydrogen ions from acid rip those bonds apart, dissolving the whole thing into solution. If the water at Jezero had been acidic enough to cause iron reduction without life, that olivine shouldn't exist. It would have dissolved billions of years ago. But it's still there, intact, which means the water wasn't strongly acidic, which means the chemical explanations don't work. One by one, the non-biological hypotheses collapsed. Now, before we get too excited, NASA has been here before and gotten burned. 1976, the Viking lander. NASA landed a probe on Mars with a biology experiment. The idea was simple. Add nutrient solution containing radioactive carbon-14 to Martian soil. If microbes were present, they'd metabolize it and release radioactive carbon dioxide gas. They ran the test. The gas appeared. Scientists were stunned. Some thought they'd confirmed alien life on the first try. But decades later, we discovered Mars soil is loaded with perchlorates, highly reactive chemicals that release gas when heated. The Viking result was chemistry, not biology. A false positive. 1996, meteorite ALH 84001. A Martian rock ejected by an ancient impact was found in Antarctica. Researchers examined it under electron microscopes and found tiny tubular structures that looked eerily like fossilized bacteria. The announcement was massive. President Clinton held a press conference. The world waited. But by 2022, those structures had been explained as products of serpentinization, a mineral transformation that happens when water reacts with certain rocks. No life required. So NASA learned to be cautious, to wait for confirmation, to resist the urge to declare victory too soon. But there's a difference this time. Both previous discoveries were explained away with specific chemical processes. This one? Researchers tried to find those processes and couldn't. There's only one way to know for sure. Bring the rock home. Perseverance has already drilled a core sample from Cheyava Falls. The tube is sealed, stored inside the rover's belly. It's sitting there right now, waiting. The Mars sample return mission is supposed to retrieve it. Here's how. A lander touches down near Perseverance, collects the sample tubes, launches them into orbit around Mars. An orbiter intercepts those samples in space, captures them, begins the journey back to Earth. Total mission cost, $11 billion. Current status, indefinitely delayed. It was supposed to launch in 2033, then pushed to 2040. Now it's in budget limbo, stuck in bureaucratic hell, despite being named the top priority for planetary science by the National Academy of Sciences. Twice. Maybe this discovery changes things. Maybe it forces the funding through. Because if those leopard spots are biological, we owe it to ourselves to confirm it. If the sample comes home, scientists have specific tests ready. Test 1. Chirality. Organic molecules come in mirror image pairs. Left-handed and right-handed versions, chemically identical but structurally flipped. Life on Earth almost exclusively uses left-handed amino acids. Non-biological chemistry produces both types equally, a 50-50 mix. If Cheyava Falls shows a strong preference for one-handedness, that's a biological signature. Test 2. Isotope ratios. Carbon exists as carbon-12 and carbon-13. Both are stable, both are common, but life prefers carbon-12. It's lighter, easier to incorporate into biological molecules. So living things have a much higher ratio of C12 to C13 than non-living materials. If the Martian sample shows that same isotopic preference, it's another clue toward biology. NASA recently developed a framework called the Confidence of Life Detection Scale. 7 levels, ranging from "we found something ambiguous" to "multiple independent teams confirmed life repeatedly." Right now, Cheyava Falls probably sits around level 3 or 4. Interesting, but not definitive. If samples return and labs worldwide confirm biological origin, that jumps to level 6. Level 7 might require returning to Mars and finding the same evidence at a completely different site. Even if this turns out not to be life, it's still extraordinary. Mars is geologically frozen. Unlike Earth, it has no plate tectonics, no continental drift. Its crust has been locked in place for over 3 billion years. Earth's surface is constantly recycled. Subduction zones drag old crust into the mantle. Volcanism resurfaces continents. Erosion wipes away ancient landscapes. Mars doesn't have that. Its early history is preserved like a fossil record we can actually read. So when we study Martian geology from 3.5 billion years ago, we're essentially looking at what Earth was like at the same age, the era when life first emerged here. Mars is a time capsule, a reference point for understanding our own origins. But if the sample is biological, everything changes. If Cheyava Falls contains fossilized microbes, here's what we immediately learn. Mars was habitable far more recently than expected. This rock formed in relatively young sediment. Life could have persisted much longer than models predicted. But the deeper question is about origins. Did life start on Mars and migrate here on meteorites? Panspermia, the seeding of planets by ejected material, is scientifically plausible. Mars had liquid water earlier than Earth. Maybe biology began there first. Or did life arise independently on both planets? That would be even more profound. See, there's a term in astrobiology called the Drake equation. It estimates the number of intelligent civilizations in the galaxy using a series of probability factors. One of those factors is FL, the fraction of habitable planets that actually develop life. Right now, we only know of one planet with life, Earth. So FL could be vanishingly small. Maybe life is a 1 in a trillion fluke. But if Mars had life, 2 and it arose separately, then 2 out of 2 neighboring habitable planets developed biology. Suddenly, FL is close to 1. Life isn't rare, it's common. And if it happened twice in one solar system, how many times has it happened across the galaxy? There's a concept called the 0-1 infinity rule. It states that in nature, things exist in quantities of 0, 1, or effectively infinite. We know life exists in at least one place. Us. If it's only one, if we're alone, then we're an accident. A statistical anomaly in an otherwise sterile universe. But if it's 2, then why not 3? 100? A billion? If life emerged twice in the same stellar neighborhood, then the universe isn't empty. It's probably teeming with biology. Most of it microbial, some of it perhaps far more complex. That's the weight of this discovery. It's not just about Mars. It's about redefining our place in existence. So, have we found life on Mars? Not yet. Not definitively. But we've found something we can't easily explain away. Something that looks biological and resists every alternative explanation we've thrown at it. And whether those leopard spots are fossils, or just geology doing something unexpected, the search itself matters. Because we're not content with mystery. We're driven to know. One day, maybe soon, we'll have our answer. Thanks for watching. If you enjoyed this breakdown, hit subscribe. We're covering every major space discovery as it happens. Drop a comment with your take. Do you think it's life, or are we reading too much into Martian chemistry? Until next time, keep questioning everything.